The Type 2 Diabetes Knowledge Portal aggregates, analyzes, and displays genetic and genomic results with the aim of helping researchers better understand this complex disease. This video will cover a set of interactive tools, the aggregation tests, that let you securely access individual-level association data to generate gene-level genetic association scores for a range of phenotypes. To use the aggregation tests, start by going to any gene page. Access the aggregation tests via the Custom Aggregation Tests link on the High Impact Variants tab. This video will give you a very quick overview, but to see the full details and documentation for the tests, download our aggregation test guide. The aggregation tests access individual-level data in the T2D-KP database in a secure manner so that the data are used in the custom analysis but never exposed publicly. If you choose to run aggregation tests for type 2 diabetes, the tests are based on the AMP T2D genes exome sequence analysis dataset, which includes T2D associations for over 49,000 exome sequences. If you choose other phenotypes, the tests access a subset of that dataset that includes about 19,000 exome sequences. First, choose a method. The differences between the methods and considerations for choosing between them are described in detail in the aggregation test guide. We'll demonstrate the interface using the first method, the additive burden test. Next, choose a phenotype. The test will compute a p-value representing the significance of the association between the variants within the gene whose page you're viewing and the phenotype you've chosen. We'll choose T2D for this example. The first two tests on the menu, the additive and collapsing burden tests, also give you the option to stratify by ancestry if desired. After setting these parameters, the next option is to select which variants go into the test. All 5 of the methods offer this option. The default set of variants shown includes variants within the coding sequence of the gene that cause protein truncating changes to the open reading frame, or variants that cause missense mutations and have minor allele frequency less than 1%. Other filters restrict the set of variants based on the impact they may have on the encoded protein, based on several different algorithms. The filters are described in detail in the documentation. You can see that each time the filter is changed, the number of variants shown in the table also changes. You can also enter a custom threshold for minor allele frequency, which overrides the MAF threshold of the filter, and can also choose to apply the threshold across all samples or across each ancestry. Finally, you can remove specific variants from the test by unchecking them in the table. The option to filter the sample set is offered for the additive and collapsing burden tests. This section shows you the distribution of traits among the sample population. Click in any box to see the distribution for that trait. You can filter the samples by entering a value in the box and selecting less than or greater than. You can also specify an internal or external range by entering 2 values separated by a comma. You can filter by multiple criteria, and if the phenotype you've chosen is T2D, you can choose to filter cases and controls separately. This allows you to design more versatile queries. For example, you could compare leaner younger T2D cases to obese older controls. Note that if the criteria you choose result in a sample set of fewer than 100 individuals, the graphic will not display and no results will be generated in order to protect patient privacy. The final step allows you to select covariates. The ability to select covariates is available for the additive and collapsing burden tests. For the other tests, this section displays the covariates used but doesn't allow you to change them. The checked covariates are used by default. After setting all desired parameters, click the Launch Analysis button. The result will show a p-value, an odds ratio or beta, and the confidence interval. If you've analyzed associations with the dichotomous phenotype T2D, a bar graph also accompanies the results. The bar graph compares the proportion of T2D cases with 1 or more effect alleles to the proportion of T2D controls with 1 or more effect alleles. If you have chosen to stratify by ancestry, the test will generate separate results for each ancestry along with meta-analysis results. The aggregation tests are under active development, and we are very interested in your feedback. Please contact us with any questions or suggestions.